Hello and welcome to a video tutorial on autosomal linkage. This is part of the unit Inheritance in A-level biology, and in the video I'm going to go over why autosomal linkage happens, and also an exam style question which actually puts this into an inheritance scenario, and why the phenotypic and genotypic expected ratios may be altered due to autosomal linkage. Okay, so firstly we're going to look at the definition of autosomal linkage. An autosome being any set of chromosomes that's not the sex chromosomes, because sex linkage is something else, and linkage being, we've got the definition here, two alleles carried on the same chromosome that are of different genes. So we're going to put that into an example over here. We've got a parent with a genotype of big A, big B, little b on the two chromosomes if autosomal linkage took place. Therefore we have a dominant allele here and a dominant allele but of a different gene on the same chromosome, and the same but for the recessive genes. So if we actually find out what the gametes of this individual would be, we would have to take it through meiosis. So that's what we're going to do. The chromosomes duplicate as we've got here, and then they split on the metaphysic plate. So that's as a result of meiosis 1, and then at the end of meiosis 2 they split again into the gametes. So I'm not going to go into meiosis in too much detail, but that's what we get. So as we can see, the only two variations of the gametes is a big A and a big B, and a little a and a little b. So as we can see here, the normal set of gametes that we'd get from an individual with this genotype would be a big A, a big B, a big A, a little b, a little a big B, and a little a and a little b. But yet here we've only got big A, big B, and little a, little b. So this is how autosomal linkage does what it does. Having the genes on the same chromosome means that we only get this. So to put that into words, so when fertilization occurs, mainly A and B, big A and big A, and big B, and little a and little b are used in that parent's gametes, as we've just seen. So we've got our big A, little a, big B, little b genotype for the parent. As we can see, if A and B, this A and B, and this A and B are linked, we get lots of those in the gametes, in the genotype of the gamete, whereas we only get little amounts of the other variations. And you're probably thinking, why do we actually still get that small amount when all that is produced here is big A, big B, little a, little b? And that comes to another part of meiosis that we look at in AS, and that's crossing over. So on the metaphysic plate, on the equator of the cell, genetic material in meiosis 1 crosses over. This is called chiasmata, and as we can see here, an example is that the big B and the little b, they cross over. So as a result of meiosis and the crossing over, this is what's formed. So that's why we still get small numbers of these two, given that these are linked and these two are linked. But that just gives a an explanation as to why autosomal linkage in meiosis only gives us these gametes, even though if there was no autosomal linkage taking place, we would expect something different, given that that's the parent genotype. Okay, so we're going to look at an example here. We're going to use the example of leaves. So the first part of this question really is not going to be much to do with autosomal linkage. So we've got two genes, a dominant and a recessive, so glossy leaves and non-glossy leaves, and then a dominant and recessive for the gene 2, which would be branching ears and no branching. So the question would be something along the lines of, given that the parent genotypes is this and this, what would be the expected ratio? So this is given that there is no autosomal or no assumed autosomal linkage. What would be the expected ratio? So you follow the normal protocol, if this was a question, you draw out your potential gametes, you draw the genetic diagram, and then you draw out your genotypes that you've got from this. And at the end of this question, and I'm not going to go into it in too much detail as this tutorial is more about autosomal linkage, you end up getting a 1:1:1:1 ratio. And that should be standard, and that's normally the same if you've got a heterozygous and a homozygous recessive both. You normally get a 1:1:1:1 ratio. And then the autosomal part of the question would be that actual results is this. So as we can see, the big G, big B, little b, and the little g, little b have got a large number of offsprings with that genotype. In fact, there are a larger number of offsprings with this genotype than there are of these two genotypes. And you may have noticed it already, but they're the same genotypes as the parents. The parents' genotypes are presented to be the most abundant genotypes in the offspring, and then this is the first sign of autosomal linkage. If you see this, if you see that there are large numbers of genotypes that are the same as the parents' genotypes, you know you're working with autosomal linkage. So this actual given ratio is 1.7 to 1.1 to 1 to 1.8. And you don't need a statistical test to tell you that that is not the same as the expected. In fact, that is quite significantly different. And then the question then would be, suggest why the expected and actual results differ. And this is a fairly standard way of answering a question like this. So I'm going to take you through it. So GB, big G, big B, and little g alleles may have been linked. And if we go back to the first part of the tutorial, that is very much like what we had here. Big A, little b, and little b were linked. So this is a similar scenario, meaning the big G, big B parent produced mostly big G, big B, and little g gametes, which again, coming back to the first slide, given that they were linked, the offspring without any crossing over only gave us those. So this is very similar, just a change of letter. Making big G, big B, little b, and homozygous recessive genotypes more common in offspring. As a result, a higher proportion of offspring have their parents' genotype phenotypes than the 1:1:1 expected ratio, given no autosomal linkage took place. So that is fairly standard. So if you wanted to maybe take a screenshot of that, that's quite helpful, and that could range from 3 to 5 marks depending on how much detail they require you to go into. But yes, linking that back to the first question, to the first slide, is that if the big A and the big B are linked, then the little a and the little b are linked, then the only gametes that you get without chiasmata or crossing over is the big A, big B, and little b. So that's a fairly standard autosomal linkage question. So it's good to remember that kind of answer. That's the key points as to why autosomal linkage and when it takes place, and why autosomal linkage means that your expected phenotypic ratios may be distorted. So I hope that helped.